Hi there, micro students. Just wanted to make a quick video. Since this is the first lab that we're doing using the Carolina Biological Kit, I thought it'd be helpful to make a general introductory video about how you should go about completing these Carolina Biological labs. What I am showing you is the page dedicated to the first lab that's entitled Aseptic Technique. And what you can see here, these are two links. The first is a handout that gives you step-by-step instructions of the lab procedure that you're going to be completing. So it would be helpful to look at that. And then also, this is the assignment that you're ultimately going to take to complete and then turn in. With all of these labs, realize that it's going to take several days, if not longer. Like this one says, it's going to take 7 days to complete. And this is due to the fact that we are going to be incubating bacteria and they need some time to grow. Since you're going to be growing these at room temperature in your household, it's going to take several days. One other thing that's really important is on this page, I give you notes. And so there are, we're going to be completing several labs. And so the bacteria that you're going to work with for this lab, it's called Serratia. We're going to use that again in the next lab. So do not discard your Serratia when you're done with this because you're going to need it in the future for the next lab. So that's some general information that I have at the beginning. And then what I have is basically a day-by-day progression of how this lab is going to look. And so what I have here is preparing for the first activity. And so these are videos that there's a combination of videos that I've put together as well as come from Carolina Biological about different procedures that you're going to be using during this. So for example, how to make a surface disinfectant, because it's important to disinfect your workspace before you start as well as after you kind of complete an activity for the day. So making a disinfectant, using tools. And so these are general videos. None of them are too long. There may be a minute or so each, minute or two. And so I've put over here the rough timeline of what you would need to set aside to complete this. Okay, so there's a series of videos. Then there is, of course, the worksheet, the assignment that I have up here. There are some pre-lab questions, and this is really just to get you to think about some of the important steps. And so before you do the activity, you're supposed to read kind of the procedure and to kind of help you focus on some of the important things. I have some pre-lab questions embedded in that assignment sheet. And so those are most helpful if you do them before you do the activity. So the first activity, so in the handout, it says activity 1. What you're going to do is reactivate one of the cultures that you're getting from the Carolina Biological Kit. So you're going to receive 4 bacteria, and currently they are what's called lyophilized. That means freeze-dried. And so we need to activate them so that they are actively growing. And so the procedure is called aseptic reactivation of the lyophilized culture. So this video shows you the generic procedure. Make sure you're selecting the correct bacteria. And so the one that we're going to work with first is called Serratia. And so the 4 bacteria in total, there's Serratia, Micrococcus, E. coli, and Staphylococcus. So you want to make sure you're getting the right one, which is Serratia. And when you follow, this link is a video that I've put together of how I reactivate the culture. So it's also the same video that's up here. It says activation of aseptic reactivation of lyophilized culture. And so when you perform that procedure, it's maybe going to take you 10 or 15 minutes to complete that. So make sure you do that, because that bacteria needs to incubate for 2 to 3 days before you can do the next step. And so what you're looking for is, and I'll show this in future videos as well, it's something called cloudiness or turbidity. And so once you have the Serratia grown, it's only at that time that you can proceed with the next activities, which are 2, 3, should be 2, 3, and 4. I'll modify that. And so those activities are going to use what's called aseptic technique. to basically take this Serratia culture that's growing. It's going to be growing in a broth. And so basically, can you transfer from the Serratia broth that's grown to a sterile broth? So it's a broth-to-broth transfer. This activity is using that same Serratia broth that's already grown. So can you transfer from a broth to a slant? In this case, can you transfer from that Serratia broth that's already grown to a sterile plate? So it's really introducing you to the different types of media and how each of those would be inoculated. So those are the different activities, and then those need to grow as well for roughly 2 to 3 days. It just depends. And then it's at this time that you're going to take your first set of pictures. And so the pictures, try to do a picture so that I can clearly see each of the 3 cultures that you're going to have. So you're going to have a broth tube, a slant tube, and a plate. And so it's helpful to take pictures individually. And whenever you take a picture, make sure to include a Post-it note with the date that you're taking the photo along with your name, so that I can, for each picture, I know who it is and when it was taken. So you're going to take a set of pictures at 48 hours, so 2 days after these have started to grow, and then another day after that, so at a total time of 72 hours. And again, take the pictures in the exact same way. Once you've done this, then that will allow you to complete this assignment sheet. And so you're going to embed these images that you've taken into that assignment sheet when you submit it. And remember at what I said at the beginning, the cultures of Serratia are going to be used in our next lab. So do not discard them right away. You're going to use Serratia as well as Micrococcus. So you're going to use 2 bacteria in the next activity. If you have any questions, don't hesitate to reach out. But because this is the first time that we're using the Carolina kit, I thought it'd be helpful to kind of do a general introduction of what you're supposed to do. I'll show you one other thing. This link is a handout that Carolina has put together. And so it's pretty lengthy, but it gives you an overview, and you can look at these. But this is very much tied to the outline that I have on that first page. So these are the different activities that you're going to be completing and the time roughly required to do each. There's some background reading, so that's important to kind of read and understand. And then they have pictures of the different items that you're going to be using. And so when you're finding them in your lab kit, you can kind of match them up. This is the culture that you're going to activate first. So this is the Serratia culture, not any of the other ones. And this is the media that you're going to be needing for these different activities. So the videos are embedded in this as well, and so you can get to them from here. Just a heads up, most of the videos embedded within the Carolina are from Carolina specifically, and the videos that I have in that first page, many of them I've made myself. So you really have 2 videos to show you something, the ones that I've made, as well as the ones that Carolina has kind of has made as well. And so there's the preparation for activity 1, and then you complete activity 1. And again, what this tells you is the bacteria has to grow until you can actually see some growth in that tube. And then it's at that time that you can proceed to activity 2. So you can't go right from 1 to 2. You need to let that first bacteria grow. I think that's all I wanted to show you. But again, I would just encourage you to reach out if you have any questions as you're preparing to do this first lab.